In this presentation, I'm going to be helping you to fill out your graphic organizer on DNA and heredity, which goes along with Module 6. You can locate this graphic organizer at the end of the Module 6 notes in your student handbook. This is probably one of the very most important topics that you will learn about in biology, so it's critically important that you pay careful attention to the explanations and fill out the graphic organizer as we go along. So let's start at the top of the first side. The first side is titled DNA and Heredity. You need to first of all make a note of what DNA, the letters, stand for. DNA stands for deoxyribonucleic acid. And just below that you will see a box that says DNA is one of the blank. If you will recall, back in Module 2, we learned about the core 4 organic molecules, and DNA is a nucleic acid. DNA is one of the nucleic acids, one of the core 4 organic compounds. Make sure that you write that in the space provided. DNA is important because it controls heredity, and this is also in a box already there for you. You're going to make 2 bullet-pointed notes regarding how DNA controls heredity. Heredity is, of course, the inheritance of traits. So your first bullet point should note that DNA is passed from parent to offspring. And this takes place through the sex cells, the gametes, sperm and egg. And then the second bullet point, DNA is a code for making proteins, and proteins determine your traits. That is so important for you to understand. That is what we call the central dogma of biology. It is the theme around which all other biological concepts revolve. DNA is a code for making proteins, and proteins determine your traits. So this is how your DNA determines what you look like and how you function. You will see in the upper right corner of your graphic organizer the same picture that is shown here. Where is your DNA? It's a question that many people have quite a misunderstanding about. So we'll start with the picture of the human, and as you know now, humans are made of cells. So label the cell in the space provided. Cells have a nucleus. That nucleus is enlarged. Label nucleus in the space provided. The nucleus contains chromosomes. Label chromosome in the space provided. Your chromosomes are made of DNA. Label DNA in the space provided. Listen very carefully as we go through this slide on DNA structure. You will see the same picture of DNA structure on the left side of your graphic organizer. I want you to start down at the bottom of that picture. You will see an area within the boxed area of DNA structure where there is a sugar labeled S, a phosphate labeled P, and a guanine labeled G, and these 3 have a box around them. Please label that area nucleotide. The nucleotide is the monomer or the subunit or the building block of DNA. You also learned this in Module 2. The nucleotide has 3 parts. So again, I need you to label these 3 parts. You are still down there in the bottom left area of the DNA structure diagram. Label the S, sugar, or deoxyribose. Label the P, phosphate, and label the G, nitrogen base. You will have to squeeze that in a bit. Now also notice that there are 2 chains of nucleotides making up the DNA molecule. One chain of nucleotides is on the left of the molecule, one chain of nucleotides is on the right of the molecule and they meet in the middle. This should be familiar to you because you have already built a model of DNA back in Module 2. You should still be looking at the DNA structure area of your graphic organizer. Over on the right side of the DNA structure area, you will see a twisted ladder. This is the shape of the DNA molecule. Up at the top of that twisted ladder, there is a box where you can label it, but don't label it twisted ladder. Label it with the correct term, a double helix. Now please look at the gray shaded areas on either side of the DNA structure. You will see that each of them is labeled sugar phosphate backbone. The backbone of the double helix, the sides of the ladder, so to speak, are alternating sugars and phosphates. The rungs of the DNA ladder, the steps, are the nitrogen base pairs. And there is a box where you can label that. At the top of the DNA structure, just above the A and the T, you can label base pairs. Just underneath the A and the T, you can label the bonds that connect the base pairs in the middle. These bonds are weak hydrogen bonds. So at this point on your DNA structure model, you should have labeled the term nucleotide as well as the three parts to the nucleotide, sugar, phosphate, and nitrogen base. You should have labeled the double helix, the base pairs, and the hydrogen bonds. There's one more thing to notice about the diagram of DNA structure, and that is how the nitrogen base pairs always pair up. You will see that there are A's for adenine. You should maybe write that in under an A or above an A. You will have to squeeze it in. Same thing for T, thymine. Same thing for C, cytosine. And the same thing for G, guanine. You will notice that always the A's and T's are paired together, and the C's and G's are paired together. We say that they are complementary. We do not say that they are opposites. We say that they are complementary. They fit together like puzzle pieces. You know by now that every living organism, whether it be a human, a tree, or a frog, or anything else, has DNA in its cells. So what makes your DNA different from a tree's DNA or a frog's DNA? There is a space to write this at the very bottom of your graphic organizer. The difference in DNA of humans, trees, and frogs is the sequence of the nitrogen base pairs. We all have DNA made of nucleotides. We all have A's and T's and C's and G's. The only difference in the DNA is the sequence of those nitrogen base pairs. Humans have about 3 billion base pairs. So it's easy to see how no two humans would have exactly the same sequence unless they're identical twins. We're now going to go over how DNA makes a copy of itself. This is called DNA replication. You have an area for DNA replication on your graphic organizer. Please go ahead and write just underneath the words DNA replication. DNA must make a copy of itself before a cell divides, and this happens during interphase of the cell cycle. You can write that in right at the top beneath the words DNA replication. It would be very helpful for you to have 2 different colored highlighters right now so that you could make sure that you color code this in order to understand the process of DNA replication. So take your first colored highlighter and highlight the original DNA, which is already labeled, in that solid color. It doesn't matter what color you use. The second step of DNA replication occurs when the DNA strand unzips. It breaks apart. Now you will need to go ahead and write in the box, original DNA strands unzipped, and you will also need to highlight those two unzipped strands in the same color that you highlighted your original DNA. Notice on the slide how this should be done. You have not used your second color yet. Now follow the arrows. The arrow going from the first side goes to the top. The arrow from the second side goes to the bottom. So you can see how those sides are intact, and they should be colored in the same color that you have been using thus far. In the case of the slide, the same color is green. The next thing that happens is unpaired nitrogen bases that are floating around in the nucleus are going to find their complementary nitrogen base and pair up with them. So since these are new nitrogen bases, here is where you would use your second colored highlighter. Pay attention to the slide so that you will see where the second colored highlighter should be used. At the end of DNA replication, you have two identical DNA molecules. Each is half new. That would be the second color, orange on this slide. Each is half old or original. That would be your first color, or green, on this slide. You should write in that statement in the box provided, two identical DNA molecules, half new, half from original. You should also highlight them, showing that each is half new and half original. We say that this process is semi-conservative because it holds on to the original strands and builds off of them. Turn now to the back side of your graphic organizer. We are going to go into more detail about the central dogma of biology, how DNA determines your traits. If you look at the bottom of this slide, you can see the sequence. DNA leads to protein, which leads to trait. And that is also written up at the top of your graphic organizer. In the first box, you are going to make some notes. DNA is used as a code for protein synthesis. Write protein synthesis in the space provided. You know by now that synthesis means to make. So protein synthesis is making proteins. The next thing that you will write in is one gene, one protein. Here's what that means. In the space labeled gene, you are going to write the definition. A sequence of DNA that codes for a particular protein. So each sequence of DNA, or gene, codes for the production of one particular protein, and your protein determines your traits. Let's look very, very carefully at the process of protein synthesis. First thing I want you to notice is the label that is already written in for you that says nuclear membrane. Find that on your paper. What this shows is on the left of that label, you are looking inside the nucleus of the cell. And on the right of that label, you are looking outside the nucleus of that cell. So we're going to start on the left of that label with the first step in protein synthesis. In the big box at the top, just beneath the words process of protein synthesis, write the word transcription. You also need to write the notation. This means DNA to RNA. More specifically, this means DNA to mRNA. And as we said, this is occurring in the nucleus. Now look very carefully at your picture and locate the double helix. It should be labeled DNA. You can see how it is labeled DNA on the slide. In the next picture, follow the arrow down. You will see that that DNA strand is unzipped. At least a part of it is unzipped, not the whole thing. This is where the mRNA is going to copy the DNA's code for that gene. Then the mRNA, which is single-stranded, is going to break away from the DNA. So at the third picture in the nucleus, follow the arrows, you will see a place to label the mature mRNA, and it's okay if you just label that mRNA. M stands for messenger RNA. It's very important that you know that. So the reason that this has happened is that DNA is a large molecule. It cannot leave the nucleus, but the code, the gene, needs to get out of the nucleus and go into the cytoplasm where the protein can be produced. So if you follow the arrow, you will see transport to cytoplasm. You will see the mRNA also in the cytoplasm. And now we're going to look at the second step. So in the large box, just on the outside to the right of the nuclear membrane, you will label translation. You will also indicate that this is mRNA to protein, and this is occurring at the ribosome in the cytoplasm. So now, look down at your mRNA strand. It's labeled, and you will see that there is a oval-shaped, egg-looking object that is not labeled. This represents the ribosome. There is a space to label it. Please do so now. So the ribosome is what essentially reads the mRNA. You will notice that attached to the ribosome, attached to 3 of the nitrogen bases, is another oddly shaped molecule. This oddly shaped molecule is called tRNA. T stands for transfer. There is a place to label the tRNA. It is actually at the one that is just beneath where you labeled the word translation. You can see that the tRNA has 3 nitrogen bases, which are called an anticodon, which is going to be complementary to a certain mRNA codon. 3 nitrogen bases on the mRNA are called a codon. Codon is kind of like word. On the other side of the tRNA is a little dot, which represents an amino acid, and the amino acids are the building blocks of protein. So the last thing that you are going to label on here is where there is a long chain of dots. You will see that long chain of dots should be labeled amino acid chain and in parentheses protein, because when it breaks away, the amino acid chain is going to fold up and become a 3-dimensional protein. We're now going to look at how we can start with a sequence of DNA and carry it through to determine the amino acid sequence that is coded for. This is on your graphic organizer. Your sequence of DNA is already written in. Your sequence of DNA is TAC, TCA, CGT, GTC. A CT. So these letters, of course, are the nitrogen bases, and they stand for thymine, adenine, cytosine, and guanine. Now, looking at the RNA line, you are going to match up the complement to the DNA bases. But recall that in RNA, there is no T. There is a U instead. So your first RNA codon will be A, U, G. T matches with A now matches with U, C matches with G. The second one, see if you can do it, should be A, G, U. The third, G, C, A. The fourth, C, A, G. And the last, U, G, A. We can then take those RNA codons and a codon chart and determine the amino acid sequence. Look carefully at the codon chart. You are first going to locate the first letter in the codon. The first letter in the RNA codon is A. The second letter in the RNA codon is U. The third letter in the RNA codon is G. So if you look carefully at the codon chart, those three letters match up at the amino acid methionine. You can abbreviate this with the first three letters, MET. MET is your first amino acid. Your second amino acid will correspond to the codon A, G, U. A, G, U is serine, S-E-R. Please make sure you're able to read the chart. Your next amino acid will correspond to the codon G, C, A. G, C, A corresponds to alanine, A-L-A. Your next amino acid will be determined by the codon C, A, G. C, A, G corresponds with glutamine, G-L-U. Your last amino acid is determined by the RNA codon U-G-A. U-G-A says stop. It's actually not an amino acid. It is an indication to the cell that the protein is finished and production should be stopped. So write in the word stop. So to summarize this process, you need to remember that there are three types of RNA. You need to write those three types of RNA in the space provided on your graphic organizer. mRNA stands for messenger RNA, and it copies a gene from the DNA. tRNA stands for transfer RNA, and it brings amino acids to the ribosomes based on the mRNA codons. rRNA is one that is often overlooked. It stands for ribosomal RNA, and it actually makes up the ribosome. Now, sometimes protein synthesis goes wrong and a mistake is made. This is called a mutation. Please, in the space provided just to the right of the word mutation, write in a definition. A change in DNA that occurs when DNA is being copied, and it may be spontaneous, meaning there's really no cause, or a result of environmental factors, for example, UV radiation, which are called mutagens. Mutagens cause mutations. There are two types of mutations, and you have a space to define each of them. In a point mutation, one single base pair is changed. So for example, the letter A might have spontaneously changed to the letter C. So this means that at most, one amino acid in the protein could be different. Now this can cause major problems. A disease called sickle cell anemia is due to a single point mutation. So here's an analogy. If you say the sentence, "The dog ate the cat," that means one thing. If you change one letter, it becomes, "The dog ate the car," and it is a completely different sentence with a completely different meaning. A frame shift mutation is an even bigger deal. In a frame shift mutation, one base pair is either added in or one base pair is deleted. So that means that from that point forward, the entire amino acid sequence will be different. There are changes from that point forward. Make sure you write that in. So this has a much larger impact. Don't forget also that sometimes mutations can be beneficial. They don't have to be harmful.